welcome back to Bioinfo. In today's explainer, we're zooming straight into the microscopic code of human individuality. When we strip away all the noise and look directly at our genomic data, the actual differences that define us are astonishingly small. Let's get right into it.
Think about this for a second. With the exception of identical twins, no two human genomes are exactly alike. Imagine looking at two massive, incredibly complex streams of genetic data flowing in perfect parallel. For literally millions of data points, they are completely indistinguishable. But then, a single base position changes. That one microscopic pivot alters everything. It shapes our traits, our phenotypes, and even our unseen vulnerabilities. But here is the wild part. That massive universe of variance, it's contained within just 0.1% of our DNA. To put that into perspective, that translates to roughly one nucleotide difference for every 1,000 bases. So when you apply that to the vast scale of the entire human genome, this tiny fraction represents millions of potential variations. It's quite literally the architectural blueprint for all human diversity. We call these single base changes single nucleotide polymorphisms, or just SNPs for short. Now, while a vast majority of these SNPs just sit in genetically silent, non-critical regions doing their own thing, there is a tiny hyperspecific subset that is responsible for almost all heritable human individuality. We're tracking millions of these known SNPs mapped out across populations because they are the defining physical markers of our biological uniqueness. Let's look at how this actually works at a specific locus. Imagine a spot where a standard reference genome holds an AT nucleotide pair. Well, a variant genome might present a GC pair in that exact same position. It's a literal single-letter swap in the biological alphabet. Identifying that solitary mismatch among billions of base pairs is, without a doubt, the ultimate needle-in-a-haystack computational challenge. So how do we actually find these hidden variations? Bioinformaticians rely on a pretty rigorous computational pipeline. First up, the raw data goes through base calling and quality control, where algorithms assign Phred scores to every read, often using tools like LongQC to ensure the data is solid. Next, filtering tools like Canoe or Prowler trim away the leftover adapter sequences. Then, heavy-hitting aligners like BWA, MEM, or Minimap2 map those high-quality reads directly against a reference sequence. Finally, all that raw code is translated into visualized, actionable data in a genome viewer, like IGV. Once a variant is successfully called and verified, it gets permanently cataloged in major databases, like NCBI's DBSNP. Take human beta-globin variants, for example. Every single validated mutation receives a unique identifier, an RSID, like RS17656961. This RSID is incredibly important. It acts as a permanent searchable coordinate, which allows researchers all over the globe to track exactly which alleles appear across different populations. But let's be real, a sequence change on its own is really just raw data. The ultimate goal is to understand the clinical consequence of that change. That's where specialized knowledge bases come in. Places like OMEM, HGMD, and PharmGKB bridge that gap. They take a specific RSID and ask the big questions. Does this substitution cause a known disease? Does it alter how a patient metabolizes a specific drug? Translating a raw variant into this kind of actionable data is the absolute core of modern targeted medicine. Now let's zoom back out to the macro scale for a minute. Global collaborations like the massive HapMap project have successfully mapped over 3.1 million of these SNPs across completely diverse populations worldwide. And researchers aren't just finding individual SNPs in isolation anymore, they're actively tracking linkage disequilibrium. Basically, they're mapping out entire haplotype blocks of alleles that are inherited together through generations. Because these SNPs occur at such an incredibly high density, they act as highly reliable physical milestones. This is the exact engine powering genome-wide association studies, or GWAS. By tracking those genetic haplotype blocks through huge populations, researchers can pinpoint complex disease risks, things like cardiovascular vulnerabilities, literally years before any physical symptoms ever appear. From linkage maps to tracking population ancestry, these single nucleotide changes are the very foundation of personal genomics. It is absolutely fascinating to think that a microscopic variance of just 0.1% acts as the true engine for all human diversity, vulnerability, and resilience. Every algorithm we run and every database we've built is specifically designed to decode that tiny fraction of a percent. As we continue to unravel the kinetic streams of our own biology, it really leaves us with one massive question. What exactly is hiding in your 0.1%?